Assalamu alaikum dear students, welcome to the course Principle in Animal Life 1. This is lecture number 53. The topic is Introduction to Cell. Discussing agenda for the topic will be eukaryotic cells have internal membranes that compartmentalize their functions, comparing prokaryotic and eukaryotic cells, and finally a panoramic view of the eukaryotic cell. Lecture outcome. After watching and listening this lecture, students will have an overview of the cell and its various organelles along with a summary of their functions. Introduction. Cell is the basic unit of structure and function of all organisms. Cell is the smallest entity which exhibit the properties of life. The cell was discovered by Robert Hooke, and he was also the one who called them as cells after the word cellula, which means empty rooms. So he basically observed dead cells in cork, which is a plant tissue, and these cells, only the cell wall was visible. The contents or the organelles inside the cell were not there, so they were giving an impression of empty rooms. So therefore he called them cells. Now there are two types of cells, and the organisms having these two types of cells are also referred to as prokaryotes or eukaryotes. The living organisms are divided into three domains. Bacteria and archaea are the first two domains, and these organisms have prokaryotic cells. Their body is made up of prokaryotic cells, and therefore these organisms are also known as prokaryotes. Whereas the third domain is the eukarya, which is further classified into 5 kingdoms: protista, fungi, animals, and plants. So these organisms have their bodies composed of eukaryotic cells, and therefore these organisms are referred to as eukaryotes. Now the kingdom protista is an informal term referring to a diverse group of mostly unicellular eukaryotes. It's a group of organisms and they are classified into phylum Protozoa. Or you can say that all the organisms in phylum Protozoa are protists. So phylum Protozoa is also referred to as phylum Protista. Now the comparison of prokaryotic and eukaryotic cells. There are certain similarities between prokaryotes and eukaryotes or their cells. These similarities are both prokaryote and eukaryotic cells are bounded by a membrane called plasma membrane, and this membrane is selective. It is selective in nature. Some of the molecules can pass through this membrane easily, other molecules pass through this membrane with difficulty, and still other molecules cannot pass through this membrane. So this selectivity of the outermost boundary or the plasma membrane is important to maintain homeostasis inside the cell. Now inside all cells is a semi-fluid jelly-like substance called cytosol, and in cytosol the different subcellular components or organelles are suspended. So you can say that the cytosol provides a medium in which different organelles are suspended. All cells contain chromosome which carry genes in the form of DNA. DNA is the hereditary material. It controls all the activities of life of an organism, and genes are the functional units of DNA. So DNA in association with proteins, mainly histone proteins, forms discrete hereditary units called chromosomes. So chromosomes are present in all living organisms. This is yet another similarity between the prokaryotic and eukaryotic cells. And all cells have ribosome. Now ribosome is called as a universal organelle because it is present in almost every type of cells. Why it is present in every type of cell? Because it is responsible for converting the language of nucleotides in mRNA to the language of amino acids in protein, and all living organisms rely on protein for their functions. So therefore the presence of ribosome is very important along with the genetic material. Now these were the four similarities between the prokaryotic cells and eukaryotic cells: plasma membrane, cytoplasm, ribosomes, and chromosomes. These are the four similarities which are present in both types of cells. Now, what are the differences? The major difference between prokaryotic and eukaryotic cells is the location of their DNA. Now, prokaryotic cells lack membrane-bounded organelles and they lack a well-defined nucleus, although they have a genetic material like their eukaryotic counterparts. But in eukaryotes, there is a well-defined nucleus which is usually present in the center or near the center of the cell in animals, but is pushed towards one side in plants because plants have a large central vacuole. So the genetic material or the DNA or the chromosomes are present in both prokaryotes and eukaryotes, but there are differences in their location or the structure where they reside. Eukaryotes have a well-defined nucleus which houses the genetic material, whereas prokaryotes lack a well-defined nucleus. However, the nuclear or the genetic material in prokaryotes is present in a structure called nucleoid. In a eukaryotic cell, most of the DNA is in an organelle called the nucleus. So nucleus is the most distinct organelle in a cell, and it is bounded by double membrane. It is also known as the control center of the cell because it has the genetic material, and genetic material contain instructions for running all the affairs of a cell or an organism. In a prokaryotic cell, the DNA is concentrated in a region that is not membrane enclosed, called the nucleoid. Here you can see a comparison of the prokaryote and the eukaryotic cell. In fact, both of them are prokaryotic cells because they are different species of bacteria. So they have fimbriae which they use for attachment. They have a nucleoid which is an area reserved for genetic material. Of course, they have ribosomes, which are universal organelles, which are the protein workbenches, which are the protein factories, which are responsible for the translation of messenger RNA into proteins, and a cell membrane or plasma membrane which surrounds the cell, which is a selective barrier. And the animal cells lack cell wall. The plant cells have a cell wall which is made up of cellulose. The bacteria also have a cell wall, but remember that the cell wall in bacteria is made up of peptidoglycan instead of cellulose. They have another structure called glycocalyx, which is the outer coating of many prokaryotes, and of course they possesses structures for movement or locomotion, and these are flagella. Eukaryotic means true nucleus. So eukaryotes have a true nucleus, a well-defined nucleus, and prokaryotes means before nucleus. So it is a Greek term. Pro means before, and it reflects the fact that the prokaryotic cells were the first ones to evolve, and thereafter eukaryotic cells evolved. So from an evolutionary point of view, prokaryotic cells are more primitive as compared to eukaryotic cells. Now the interior of either type of cell is called the cytoplasm. In eukaryotic cells, this term refers only to the region between the nucleus and the plasma membrane. Within the cytoplasm of a eukaryotic cell, suspended in cytosol, are a variety of organelles of specialized form and function. So as already discussed, the cytosol provides a medium. It is a gel-like semi-fluid substance, and it provides a medium where the subcellular organelles are suspended, and the membrane-bounded organelles are absent in prokaryotic cells. So now we are discussing the differences between the prokaryotic and eukaryotic cells. The major difference is the prokaryotic cells lack a well-defined nucleus, whereas eukaryotic cells have a well-defined nucleus. And the second difference is the prokaryotic cells lack membrane-bounded organelles. Membrane-bounded organelles means organelles which have a membrane surrounded around them. So the ribosome is a type of organelle which does not have any membrane surrounding it. Therefore, ribosome is present both in prokaryotes and eukaryotes, whereas other structures like the Golgi bodies, the endoplasmic reticulum, and the nucleus, these structures have membranes, mitochondria. So these membrane-bounded structures are not present in prokaryotic cells. In spite of the absence of organelles, though, the prokaryotic cytoplasm is not a formless soup. Although the membrane-bounded organelles are absent in the prokaryotic cells, but their cytoplasm also have certain structures which enables them to survive. For example, some prokaryotes contain regions surrounded by proteins, not membranes, within which specific reactions takes place. So although the structures the prokaryotes possesses are not surrounded by membranes, they are surrounded by proteins, and where the vital functions of the cells takes place. Eukaryotic cells are generally much larger than prokaryotic cells, and I will give you a very good example of this, that the human body, inside the human body, there are 10 times more bacterial cells than our own cells. So it will give you a good idea about how small these prokaryotic cells are. Now, size is a general feature of cell structure that relates to function. So, at minimum, a cell must be able to house enough DNA, protein molecules, and internal structures to survive and reproduce. The maximum size of a cell is influenced by its requirement for enough surface area to obtain adequate nutrients and oxygen from the environment and dispose of wastes. Size is also limited by the distance these materials must diffuse within a cell. So the extent of metabolic activities and the function performed by a cells determine the size of the cells. The logistic of carrying out cellular metabolism sets limits on cell size. So, as already mentioned, the function performed by cells and the extent of metabolic activities that can take place inside a cell are the two variables which control or which have a considerable effect on the overall size of the cell. Now, at the lower limit, we have mycoplasmas. Mycoplasma are the smallest cells known, and of course these are bacteria, and their diameter is from 0.1 to 1 micrometer. So remember, these are the smallest cells known. These are perhaps the smallest packages with enough DNA to program metabolism and enough enzymes and other cellular equipment to carry out the activities necessary for a cell to sustain itself and reproduce. So the extent of metabolic activities and the function performed by a cell will determine the size of that cell. Typical bacteria are 1 to 5 micrometer in diameter, and that is about 10 times the size of mycoplasma. So mycoplasmas are also bacteria, but they are the smallest living cells known. And the typical or the usual bacterial cell have a diameter of 1 to 5 micrometer, which is 10 times greater than mycoplasma. And if you compare a eukaryotic cell to that size, it is 10 to 100 times greater than bacteria, typical bacteria in diameter. It is 10 to 100 micrometer in diameter, so you can say it is about 10 times greater in size than bacterial cells. Now, metabolic requirements also impose theoretical upper limits on the size that is practical for a single cell. At the boundary of every cell, the plasma membrane functions as a selective barrier that allows passage of enough oxygen, nutrients, and wastes to service the entire cell depending upon the need of the cell, the need for oxygen, the need for nutrients, and the disposal of waste material. So the surface area to that cell is provided accordingly. So there is a structure of plasma membrane, which is a lipid bilayer embedded with proteins, and the lipid bilayer is hydrophilic on one side and their tails are hydrophobic. So such type of molecule which exhibit both hydrophobic and hydrophilic characteristics are known as amphipathic molecule. So here you will find out that smaller cells have greater surface area to volume ratio as compared to larger cells. On the right side of your picture you can see a small cell where each side is 1 in length and width and breadth. Now this one could be anything. It could be a micrometer, it could be a millimeter, it could be a centimeter. Therefore they have mentioned as unit instead of putting a specific unit. And here we have a large cell where each side is 5 in length, width and breadth, and then we have a group of cells, a large number of cells, so it is the aggregation of these small cells. So here we will calculate how the size of the cell has relationship with the surface area to volume ratio. So here a single cell which is cube shaped and the cube has 6 sides. So the total surface area is calculated as height into width of one side, which is 1 into 1, and then there are 6 sides in a cube. So 1 into 6 makes 6, and then multiply by number of cells. So here we have only one cell, so we will multiply 6 with 1, it will give us 6. Now calculating the total surface area for this large single cell, each side is 5 in length, width and breadth. So height is 5 and width is 5. 5 into 5 gives us 25, and then we multiply 25 with 6, so it gives us 150, and because there is only one cell, so 150 into 1 is equal to 150. Now we calculate the surface area for this group of cells. So each cell is 1 any unit, centimeter or micrometer or millimeter. So 1 into 1 is equal to 1, and then multiply with 6 because a cube has 6 sides, so it gives you 6, and then multiply it with the number of cells. So if you multiply 6 with 125, then it will give you 750. So these are 125 cells, and that is their total surface area. Now look at the increase in the total surface area. Here of course is a large cell, so its total surface area is greater than this small cell. But if this much area is occupied by smaller cells, then their overall surface area will be greater than a single large cell, as you can see here. Now the total volume is calculated via multiplying height with width with length for each cell, and then multiplying it with the total number of cells. So here 1 into 1 into 1 multiplied by 1 is equal to 1, and for this cell we have 5 into 5, that makes 25, and then 25 into 5, that makes 125, and then because there is only one cell, so 125 into 1 is equal to 125. And now for this, a single cell has one width, length, and height. So 1 into 1 into 1 is equal to 1, and then multiply it with the number of cells, which is 125. So 1 into 125 is equal to 125. Now the important thing, the surface area to volume ratio. So how it will be calculated? You will have to divide the surface area on volume. So here we get 6. You write the surface area in numerator and then divide it by the total volume, which will be written in denominator, and then you will get 6. And here 125 divided by 150 divided by 125, so it gives you 1.2. And here 750 is the total surface area, total volume is 125, and if you divide 750 on 125, it gives you 6. So you can see here that a larger cell has lesser surface area to volume ratio as compared to smaller cells, because the smaller cells, if they have gaps in between, then there will be more area exposed to the extracellular environment as compared to a single large cell. So the smaller cells, the body is made up of smaller cells, because the smaller cells can perform their function efficiently as compared to larger cells, and they can interact with the extracellular environment efficiently as compared to larger cells. Now, the need for a surface area large enough to accommodate the volume helps explain the microscopic size of most cells. So cells are smaller in size because by having a smaller size, they can have more surface area as compared to few large cells. Large organisms do not generally have large cells than smaller organisms. They simply have more cells. So some organisms are smaller in size, others are larger in size. So this larger size of the organism is not because of having larger cells, but it is because of having more number of smaller cells as compared to the smaller organisms. And remember from the previous slide that the smaller cells have more surface area to volume ratio as compared to the larger cells. So it is more beneficial for organism to have smaller cells as compared to having larger cells. A sufficiently high ratio of surface to volume is especially important. Why? What is the importance of the surface area? Because the importance is the surface area of a cell is facing the extracellular environment where the exchange of materials takes place. So a sufficient surface area is always important for a cell. And here you have example of intestinal cells, which are flattened cells, and because they are concerned with absorption of nutrients. Such cells may have long, thin projections, and one more structure that is provided to those cells is the microvilli, which further increases the surface area. These microvilli are hair-like projections. Now, a panoramic view of the eukaryotic cell. Panoramic means a wide-ranging, a broad view. So in addition to the plasma membrane at its outer surface, a eukaryotic cell has extensive, elaborately arranged internal membranes that divide the cell into compartments. The organelle is mentioned earlier. So if you remember from the discussing agenda, we said that the cell has internal compartments, and these internal compartments are bound by membranes, and because of that different activities can take place in these internal compartments. For example, lysosomes are bound by membrane where degradation takes place. Golgi bodies are bound in membrane where the sorting, packaging and shipping of substances takes place. Endoplasmic reticulum is also bound by membrane where the synthesis of lipids takes place, where the detoxification of drugs takes place, where the synthesis of proteins takes place, and mitochondria is also bound by membrane where the process of Krebs cycle and electron transport chain takes place. So these membranes surrounding different structures, they enable these structures to carry out their distinctive roles in a diverse cellular system. The cell's compartments provide different local environments that support specific metabolic functions, so incompatible processes can occur simultaneously in a single cell. So these compartments enable the cell to carry out different types of activities which would not have been possible inside a cytoplasm. The plasma membrane and organelle membranes also participate directly in the cell's metabolism because many enzymes are built right into the membranes. The basic fabric of most biological membranes is a double layer of phospholipid and other lipids. So this is the structure of the plasma membrane, which is a lipid bilayer embedded with different kinds of proteins. Here once again you have the structure of a plasma membrane. It's a lipid bilayer and different proteins are arranged. Proteins may be intrinsic or extrinsic, integral or peripheral, and the outer surfaces of some of the proteins are supplied with cell surface markers. However, each type of membrane has a unique composition of lipid and proteins suited to that membrane's specific function. So the enzymes and different components are embedded in different membranes. For example, if you look at the membranes of the mitochondria, there the four complexes are embedded, the four different types of proteins are embedded, where the process of oxidative phosphorylation takes place, and where they reside the different electron carriers. So these specific types of proteins are of course not there in the membranes of endoplasmic reticulum or Golgi bodies. So the structure of membrane surrounding a specific organelle is specially fashioned or designed to play some role in the function performed by that organelle. For example, enzymes embedded in the membranes of the organelles called mitochondria function in cellular respiration. Now, on the next two slides, we will have a generalized diagram of plant, animal, and plant cells. So this is the structure of an animal cell. The flagellum, which is used for motility. Endoplasmic reticulum is of course divided into two types: rough endoplasmic reticulum, which is supplied with ribosomes, and smooth endoplasmic reticulum, which don't have ribosomes and they have characteristic functions. Then we have a nuclear envelope which surrounds the nucleus, a nucleolus, where ribosomes are synthesized, the smaller and larger subunits of ribosomes. Then we have a chromatin, which is the association of DNA and proteins. Of course, all the cells have plasma membrane, which is the selective barrier, which is the traffic controller. And then we have ribosomes, which are the structures responsible for translation of messenger RNA into a polypeptide chain. Then we have Golgi apparatus, which is also known as the post office of the cell, and responsible for packaging, sorting, tagging, and shipping of proteins. Then we have lysosomes, which are also known as suicidal bags because they carry enzymes. And we have mitochondria, which is the powerhouse of the cell, where the Krebs cycle and electron transport chain, the two steps of the cellular respiration takes place. Then we have peroxisomes, and these are organelles with various specialized metabolic functions. Initially, when we were discussing different organelles, we skipped this organelle, but here we will discuss it in the subsequent lectures. Then we have microvilli. Microvilli are membrane projections, and these increases the surface area for absorption. In the last slides, you saw the structures in the intestinal cells. They are projected outward, and because of that, the surface area available for absorption is increased, and therefore there are cytoskeletal elements. Of course, they are important because they are responsible for intracellular transport, and we say that the nucleus is present in the center or near the center. So it is because of these cytoskeletal elements they give a proper shape to the cell. And then the cytoskeletal elements are of three types. Then we have a centrosome, where the cell's microtubules are initiated, that is during the cell division. So this was the panoramic view of a eukaryotic cell and a eukaryotic animal cell. Now the plant cell. So the major difference here is the presence of this structure. This is the cell wall. Cell wall is present in plant cells. It is absent in animal cells. So the outermost boundary of the plant cell is cell wall, and the outermost boundary of the animal cell is cell membrane. So you can see that almost all the structures which were present in the animal cell are also there in plant cell. And in the plant cell, the nucleus is usually pushed towards one side because of the presence of a large vacuole. And then of course the plant cells carry out the process of photosynthesis. So the chloroplast is also present there in plant cells, and the plant cells are associated or in communication with adjacent cells through plasmodesmata. We usually call these structures as cell junctions in animals. So with that we come to the end of this lecture. You can refer to Campbell's book for further details. Thank you very much. Allah Hafiz.